Your research, broadly speaking, looks at the intersection between the brain and the immune system. My understanding is that that connection is relatively recent. So I wonder if you could talk a little bit about it. And so let's pretend you're talking to someone with a ninth grade understanding of science, and hypothetically, let's call this person the president of UVA. Hello, everyone. I'm Jim Ryan, president of the University of Virginia, and I'd like to welcome all of you to another episode of Inside UVA. This podcast is a chance for me to speak with some of the amazing people at the university and to learn more about what they do and who they are. My hope is that listeners will ultimately have a better understanding of how UVA works and a deeper appreciation of the remarkably talented and dedicated people who make UVA the institution it is. I'm joined today by Dr. John Lukens, Associate Professor of Neuroscience and Principal Investigator of the Lukens Laboratory. Dr. Lukens is an expert immunologist and neuroscientist. He's dedicated his career to improving our understanding of how the immune system can contribute to the development or prevention of neurological disease. His laboratory is engaged in important research around highly complex neurological diseases such as ALS, Alzheimer's, Parkinson's, multiple sclerosis, and others, helping to improve our ability to treat these diseases. Dr. Lukens received his BS from the University of Richmond and his PhD from the University of Virginia. He is an eminent researcher, scholar, and educator, as well as an outstanding mentor, husband, and father. And today we are fortunate to have him on the podcast. Dr. Lukens, thanks for being here.
Yeah, my pleasure. Excited to be here, Jim.
Is it okay if I call you John?
Yeah, if it's okay if I call you Jim.
Absolutely.
Okay.
Sir works as well.
Okay.
So I understand you're a Philly guy. Did you grow up right in the city or outside of Philadelphia?
Yeah, so we spent my first couple of years in Roxborough, which is just west of this. It's a borough in the city. And then we moved out to an area called Plymouth Whitemarsh. So it's just like a mile outside of the city.
Eagles fan, Phillies fan?
Gotta be.
I'm sorry
there's
to
no
hear
other
that.
way. No, Philly's great. The fans are passionate. It's just an amazing place to have grown up. I enjoyed every second of it, and my kids now, even though they're diehard Cavs fans, UVA. Second comes all Philly sports.
Right. Well, I'll forgive you as a Giants fan. We root for our hometown teams. So
Yeah.
did you go to the Jersey Shore as a kid?
Oh yeah, every summer. So that's where, I pretty much spent most of my family vacation. So we would go to Ocean City, and then I was actually a lifeguard in Sea City one summer.
Oh, no kidding.
Yeah. So, and then, yeah, especially during high school, we spent our summers there. I worked as a chef one year at one of the breakfast places, and a lot of fantastic memories. Going down to the Jersey Shore with my parents, extended family, and brothers and sisters. And we take our kids there. We try to go up to Philly once a year. We do a Phillies game. We usually get in a soccer game. And then we do day trips to Ocean City.
And so, am I right? You're a first generation college student?
Yes, that's correct.
And how did you end up at Richmond from Philadelphia?
Yeah, so it just so happened one of the guys that I played ice hockey with in high school went there. He was a year ahead of me, and he was somebody I looked up to. I went to advice to him about like AP Bio and AP Chemistry, and so he kind of put me onto it. And then I went there and absolutely loved it in the sense, very small classroom sizes and a real emphasis on research. And you can get involved in research like straight out of the gate. It was my first semester. I was already in the lab. Had a really strong connection with my mentor there. It was a great place. Yeah.
And when did you realize that you were interested in science? Really young? Did it happen in high school or college or?
Yeah, it happened in high school. Then I was, I just really got into biology and chemistry and loved the idea of wanting to help people get better. I just was fascinated and driven to find, treatments and novel ways to help people with devastating ailments. And that was kind of the passion. I liked kind of the problem solving and the unknown. and especially in the case of biology, looking back, there's been quite a revolution in terms of technology,
Yeah.
in genetics and big data, and it's amazing how far research has come, and just the opportunity to make a difference was kind of what drove me to science and research.
And did you go into college thinking that you wanted to get a PhD, or did that decision occur while you were in college?
Yeah, so that happened towards the end of undergrad. I went in thinking that I went to do undergraduate education,
Yeah.
and as I got into the lab and spent a couple summers in the lab, I just fell in love with the opportunity to I mean, it's just an amazing feeling to get a piece of data and realize you're might be one of the only people in the world at that moment that knows that thing. And that I
Yeah.
found that to be very, it drew me in further. And I liked, how fast paced it was, and, in terms of publishing. And I saw, I guess a better opportunity in terms of using my skills to go to research and to get, to maybe do something more in science. I actually, when I came into college, I was doing organic chemistry research, and I loved it. Like I said, my mentor, we played softball during the summer, and his name's John Gupton, but he's, he in a lot of ways he's how I kind of model my mentoring style. He was just really got to know all of his trainees and treated us like family, both, the good and the bad in the sense of pushing us to be better every
Yeah.
day, but also, putting his arm around us when, the experiments didn't work the way we wanted. And, I didn't take immunology until my last year of undergrad. And what drew me to immunology was the fact that it's really hard to find a human disease where immunology doesn't play a major role, if not
Right.
the underlying role. Everything from cancer, how the immune system helps to remove tumors, to infectious disease, autoimmune disease. And, even in the last couple of years, we've come to know that it's really critical in maintaining health of the brain too.
Right. Yeah. So I want to come back to that. Let me
Okay.
just ask you, what drew you to UVA?
Yeah, so I really connected with the PIs that I met with
Hmm.
during my interview weekend. At the time, there was a new immunology center, the Byrne Carter Immunology Center, that was kind of just getting off the ground. There were a lot of new PIs that had really exciting projects. And, kind of getting back to the Philly thing, the center director, Tom Bargiali, he grew up in South Philly, first
Okay.
generation. And I could tell that, he was really invested in me as a person and also a scientist. And it felt like home. I've always kind of approached where I went in terms of schooling, like, it's more than just where you learn facts. It's also gonna be your home for those years. And
Right.
you want to have people around you that, are really going to invest in you and your development. And definitely felt that way with Tom and the others in that new immunology center.
Yeah. So going to your research, so
Mm-hmm.
your research, broadly speaking, looks at the intersection between the brain and the immune system. And my understanding is that that connection is relatively recent. So I wonder if you could talk a little bit about it, why it took so long for people to understand the connection and why it's important. And you might, let's pretend you're talking to someone with a ninth grade understanding of science, and hypothetically, let's call this person the president of UVA.
Yeah. Yeah. So for the longest time, the kind of prevailing paradigm has been that the brain is an immune privileged organ where it's separated from the immune response and there's very little communication. And the thought was, and, I guess conceptually, you could make a strong argument. You wouldn't want an immune response to be overactivated in the brain because you have these neurons that are important for storing memories and also for controlling our movements and pretty much our everyday life. They're postmitotic, meaning that they're not coming back. They're not going to be replaced. They're not like our
See.
skin cells. that when you get an injury or you lose some, you get a replacement. So this was kind of a simplistic idea. And there was also the fact that there was this fortress around the brain, which is called the blood-brain barrier. And this
Right.
kind of was really difficult to get immune cells in. And so this, lasted up until maybe like 10 to 20 years ago. And probably some people still kind of agree with it in some ways. But the real, I guess, thing that caused this paradigm to crumble was there was three real things. One of them is advancements in neuroimaging.
Mm-hmm.
So as we begin to look closer, we realize that most neurological disorders have activation of the immune system in the brain, and that the activation of these immune cells in the brain, the more active they are, typically correlates with the severity of it and how much tissue loss has been there. And then the other thing, or the second thing that kind of led to this, was advancements in genetics. As the technology's gotten better and it's gotten cheaper to do large scale genetic screens, we've come to realize that many of the genetic risk factors for things that we thought were purely neuronal were actually targeting the immune system. So if you look at Alzheimer's disease, the genome-wide association studies that have come out in the last 15 years, most of the genes are immune genes or genes that are almost exclusively expressed by the one immune cell that resides in the brain. And this is kind of changed our thoughts on a potential role for the immune system. And then the third thing is, something that was kind of came out of UVA, actually. And it's this idea of the role of the meninges. So there's this really thin membrane above our brain between our skull and the brain parenchyma. And typically, when people did autopsies, they would cut the skulls off the brains and then peel it off. And what was peeled off, sticking to the skull, which they threw in the trash can, was the meninges. And it's this, what we've come to know through discoveries that were made here at UVA, is that this meningeal, this small little layer that people were throwing out in their autopsies, is just filled with a massive composition of immune cells. And these immune cells can release factors that can coordinate the neurons to do certain things, and to affect memory and anxiety and mood. And then the other thing was something that happened, I guess it was probably 8 years ago while I was still here. It was discovered that the brain had lymphatics. So this drainage pathway that most other organs have, and the brain was thought not to have. And that would kind of added to the immune privilege idea that was being kind of touted around. And this is the major way that you get waste out of organs.
Right.
But it's also the communication between the organ and the immune system.
Right.
So those three things were what kind of tore down this incorrect paradigm. And it's been pretty recent, and it's exciting in that sense because there's so much opportunity now.
Yeah. So talk a little bit about that. I mean, what does this open up? I mean, it's obviously an incredibly interesting thing to discover something fundamental about the brain and the immune system. But now that we know there is a connection and they're not completely separate, why is that significant? And what other questions are raised by that?
Yes. So there's multiple things. So with the communication now, we know that we can take basically the fluid that's being drained through those lymphatics, and we can profile it. And we can say, are there neurotoxic material in there? How are the immune cells? And one of the biggest things, the limitations to treating a lot of neurological diseases, is we don't have good biomarkers to say, let's intervene now. And so most of the times when people have tried to intervene, it's been too late, in the sense that what's lost already isn't going to come back. And this provides an opportunity to really, it's like a window into the brain where you don't have to be as invasive to go in and sample.
Right.
And so that's been one thing. And then the immune system and the meninges are incredibly targetable, in the sense that it's really hard to get things across the blood-brain barrier. So that's a limitation. But in the immune system, being in the periphery, it's much easier to target it. And we've had, decades of advancements in terms of immunomodulatory drugs and treatments. I mean, things like psoriasis that's targeting the immune system. If you look at multiple sclerosis, all of the drugs that are available for multiple sclerosis are targeting the immune system. So we kind of have a toolbox that we could deploy almost immediately. And it's a little bit less dangerous than, say, like perturbing a neuron, whereas you can kind of fine tune the immune response, and you don't have to worry about as much collateral damage of causing the neuron to burst or whatever.
So when you say targeting the immune system, this may be a silly question, but am I right that the problem with the immune system is that sometimes when it's reacting to something, it actually causes harm rather than, because I think a layperson, including myself, would think it's really good that we have an immune system. It fights off colds. But is it the case that sometimes in the process of fighting off something, it actually causes more damage?
Yeah, that's correct. Immune system in pretty much every organ now is always a balance between the beneficial versus the detrimental. And, this is really well appreciated in terms of viral infections of the lungs, where if you get too robust of an immune response, your immune system can cause too much damage as it's trying to clear the virus. And then you obviously have breakdown in lung function, and it's immunopathology that's driving a lot of the issues. But, on the other hand, you need your immune system there for tissue repair. And also to kind of corral and contain anything that's a danger, whether that's an infection or say something neurotoxic. So things like amyloid beta or alpha-synuclein, which are the two drivers of Alzheimer's disease and Parkinson's disease. So it's really harnessing the good and the bad to find the sweet spot to unleash it in a beneficial way that is the real sweet spot there.
Right. So let me ask you a silly question. Why doesn't our immune system work better?
Yeah, no, it's a great question. So with time, the biggest problem is with time, the immune system kind of breaks down, and this occurs, yeah. And so basically they almost become functionally exhausted, or they get overloaded with neuronal debris that's bad. And then they're not as good at clearing the material, and then the alarm bells ring, and they start releasing other factors that aren't pro-inflammatory. So with a lot of the aging associated neurodegenerative disease, they reach a state of overcommitment, and basically they can't keep up with the pathology. So you kind of need ways to rejuvenate them. And that's some of the things that we've been working on. And a similar thing kind of is present in cancer, whereas the immune system is really good at controlling cancers early on, and then it becomes functionally exhausted. And it still knows that there's a tumor cell there, and it can engage with it, but it can't release the payload that would kill that tumor.
Interesting.
And so that concept of rejuvenating that functional exhaustion is also what our lab and others are trying to do in terms of rejuvenating the normal beneficial functions of, say, the cells in the brain that typically protect our brains.
Talk a little bit about what you're working on now and any significant recent discoveries you've made in your lab.
Yeah. So what we're working on now is we're really trying to understand what are the brakes and accelerators to turn up or turn down these microglia. So these are the only immune cells that reside in the brain. And these are the cells that contain a lot of the risk factors for most neurodegenerative diseases. And we've been trying to identify various molecular factors that can boost their beneficial functions as well as turn them off. So work that was done by Hannah Ennerfelt, who was a grad student in the lab. She's now moved on to do a postdoc at Stanford. She identified this molecule SYK, spleen tyrosine kinase. And it seems to be a major hub that coordinates neuroprotective functions of the microglia. And she's shown by targeting it and over activating it with various means, you can improve the removal of amyloid beta and limit Alzheimer's related disease. She's also shown it in a model of multiple sclerosis too. And so we have various targets that we're going after in hopes of identifying multiple things that can move forward into translational tests and hopeful therapeutics in the next 10 to 20 years.
So that seems pretty significant, that discovery.
We were super excited about it. It was a fantastic project. It was ambitious and a little daunting, but it was amazing, the perseverance and kind of all the effort that Hannah put into it to take it across the finish line. You submit these papers and you get a ton of things back in the review process where they're critiquing how you can make it better and not. And it was a really exciting time. And I still remember like the first time she came in and showed me the images of the amyloid levels and just the pure excitement that comes with that, to see that eureka moment. And it's awesome.
I bet. I mean, you're basically getting a look at what might be a mechanism that leads to Alzheimer's or could lead to intervening to mitigate or cure Alzheimer's.
Yeah, exactly. And it's been interesting to we've kind of branched out from her initial studies. Work we're doing with Bill Petrie in NeuroCovid suggests that this same sick molecule that is involved in Alzheimer's disease is also acting up in a dysregulated fashion in microglia in NeuroCovid, at least in some of our early studies. So we're also working with Taji Harris down the lab. She's looking at it in terms of a parasitic infection of the brain. And it's been kind of exciting to see how focal this one pathway is in terms of orchestrating these important immune cells in the brain.
So when you're in the course of discovering these mechanisms in this molecule, do you have a sense of how far down the path you are? Like, do you have a sense of, gosh, we are just knocking at the door to figuring out the biomechanics of Alzheimer's, or no idea if we're close or not? We don't know enough yet to know we are really close to making a huge breakthrough. Does that question even make sense?
Yeah, no, it definitely makes sense. I think the way that we approach it is that, once we kind of get a center point in the pathway, we immediately try to go upstream or downstream of it to identify what is other factors are coordinating it. And our thought there is not always the first molecule that you figure out that's important in the pathway is gonna be a good drug target. So we might find out by, targeting it everywhere, it might play an important role, say, in the kidneys. But by going upstream and downstream, we can hit on the unique players that only hitting them could still bring about the activation of, say, sick, the beneficial, without causing any issues or collateral damage in any beneficial roles that it would play. So you don't necessarily know it's like a slam dunk right away, because just like the discovery, there's a lot of unknowns and surprises in biology once you start targeting it globally.
Right. So you're the principal investigator, so that means you lead a lab. Can you describe for folks what it's like to lead a lab? I mean, this is a team sport, right?
Yeah, no, it's in my opinion, it's the best job ever. And you said team sport, and I kind of, I approach it kind of like a coach in the sense that, every day, trying to help people and the trainees with identifying their strengths and weaknesses and building on those. So, some of the trainees come in with really fantastic creative ideas, but really need to refine their surgical technique. And others come in as fantastic writers, but don't have at the time, the confidence to really think outside the box. And so, and it's nice because everybody has their different strengths and weaknesses, and we can kind of pull from each of those to have a bigger effect as a collective. And it's a lot of fun to see. It's also, it's even more fun to see them, move on to bigger and better
Right.
things. Yeah, I mean, it's just amazing where some of these trainees have gone, leaving UVA, and hopefully we can get a lot of them back as
Right.
faculty or as doctors, because they're just incredible.
And so you have a mix of undergraduate, graduate students, and postdocs.
Yeah, so right now about 8 undergraduates, 8 graduate students.
Wow.
Some of those individuals are co-mentored. So one of our trainees is co-mentored with Bill Petrie on
Mhm.
NeuroCovid, and then we have another co-mentored student with Rich Price, where we're trying to develop new therapeutics to target some of the pathways that we have identified. And then we have 2 postdocs, and then we also have we have 3 lab techs.
And you said that your mentor treated you like family. Is that how you try to treat the people in your lab as well?
Yeah, absolutely. Like it's so rewarding and fun to be, part of their lives. It's a unique, mentee mentor relationship in that it's like a true apprenticeship where, you see them come in and, they're just bright-eyed and excited and, want to cure various diseases, and to see them leave the lab and, have been so accomplished, and also to have had a lot of fun, and to make huge discoveries and get recognized for that is just, there's nothing better than seeing your people do well.
So just the last couple of questions. I
Uh-huh.
understand you have a spirit week in your
Yeah.
lab. What does spirit week in your lab look like?
Yeah, so this started a couple of years ago. It's basically like your high school spirit week. So, some of the days like pajama day, wacky hair day. And the one day they got me in the sense that it was like, what my midlife crisis right now is Air Jordans and Nikes. So they said like it was going to be like fire shoes day, like, shoes that were really cool. And I came in and I saw the first student and I was like, oh, your shoes are okay, but you look really dapper today. And then I realized that they were all dressed like me. So
So
they had,
it was Dr. Lukens Day.
yeah, yeah, yeah. And then the pajama day, that we didn't think ahead. So there was a public defense in our department. One of the other lab students was getting their PhD, and this is a pretty formal affair.
Yeah.
And we wanted to be there to support it, but we completely forgot that it overlapped with our pajama day. So we showed up as a whole lab in pajamas, and that's kind of become a tradition where now whoever's graduating gets to decide what we dress up as a lab. It's kind of been fun that way.
Last question about your sneakers. So I
Mhm.
understand you've developed a tradition around research publications that involves your sneakers.
Yeah, it kind of I don't know. It's so much work goes into these papers and grants. I feel like it's important to like reflect on getting across the finish line. And it started a couple years ago. Like I said, it's my midlife crisis. There could be worse things. But yeah, so when I got tenure, I got the UVA Dunks, which were
Oh,
the
nice.
the shoes they made when we won the national championship. And then I try to get something that like kind of reflects the work that was done. So when we published the sick paper, it became a joke. There's a song out there by Travis Scott and Drake called "Sicko Mode," and yeah, Travis Scott ended up making a pair of Jordans. So I ended up getting those to kind of remember sick and the time that went into that. Yeah, it buys me a lot of street cred with the students.
Uh, well, John, thank you so much
Yeah,
for spending some time with me. I often talk about UVA being great and good, and I usually explain what I mean with examples, and you're a perfect example of what I mean when I say UVA is great and good. So thank you for everything that you're doing, and thank you for spending time with us.
my pleasure. And thank you for that. That means a lot to me.
Inside UVA is a production of WTJU 91.1 FM and the Office of the President at the University of Virginia. Inside UVA is produced by Jaden Evans, Ariane Ballou, Mary Gardner McGee, and Matt Weber. Special thanks to Maria Jones and McGregor McCants. Our music is Turning to You from Blue Dot Sessions. You can listen and subscribe to Inside UVA on Apple Podcasts, Spotify, or wherever you get your podcasts. We'll be back soon with another conversation about the life of the university.